This is great because first I want to thank the speakers before me, because now I've got an extra 20 minutes just to say whatever I want for the rest of the day. So you guys may want to just relax, get some water, step back and relax. I want to first and thank Dr. Carrico, Dr. Ramirez, and really all the speakers. They put a ton of time into this, as I think you can all tell. And as Julio said in the beginning, each of these lectures could literally be hours long. When I was sitting listening to Stephen and Thomas, thinking I got a master's in statistics, and thinking that was 2 years of time. They shrunk down to 40 minutes. So kudos to you guys, an amazing job. So I love this slide. I think if you don't know anything else, this slide gives you the blueprint for research. And as I look at this and I think about this, the full continuum of what it means to go from the idea to the conclusion, it made me think of a study that when I was a young doctor, 1994, there was this really interesting study in New England Journal of Medicine. And most of the examples I give you are going to be cancer, because that's what I do. But so this was a lung cancer study, and it was really based on some sound biology. And they said, what we've realized is vegetarians do not get lung cancer. So they made the supposition that we're going to study beta carotene and the impact on lung cancer. And it was an exquisite study, 30,000 men in Finland, smokers. And they did this study over an extended period of time. It was a randomized, blinded study. And they did all this great work. Now, understand, as was said a couple of times, it was published in the New England Journal of Medicine. So pretty good journal. And what do you think they found? When they actually gave people beta carotene and studied very carefully. Well, guess what I'm thinking, but they actually showed that those people got beta carotene had more lung cancer. So it was the exact opposite of what the premise was. And as we went through that in those days, I was really thinking about that extensively. Why is that? Is it something in this chart that you see that they misstepped? No, it's really about what we heard earlier about confounders, about the fact that the biology is complicated. So we try to simplify things. And we all saw it during COVID, right? Somebody stepped in a banana peel and they got COVID, went away, and it was like, everyone step on banana peels quick, right? And it's that kind of thing. Our job is to look at it much differently. So, so what's clinical analysis, right? What's the analysis part of it? I'd be remiss not to, I have tons of statistician jokes, but I'm going to save those for today. But I do, my dad was in the liquor business. And when I was going through getting my master's, we always talk. And so he was in the liquor business, and he used to say, Joey, you know, more people die after leaving a doctor than all the ones who leave a bar. And I was thinking, and like, well, that's statistics. It's right. I'm not suggesting people go out drinking instead of going to their doctor, but yeah, because they're in the hospital. But Dad, they're in the hospital. The point being is that the analysis part is taking that statistics and putting some sense around that. Okay. So as we go through this in 20 minutes or less, we're going to look at consistency, the strength of the data and the association, temporality, the biology. Biology is important because that really goes back to that little joke I just told about how do we deal with that. Generalizability, and I think really, it was said a couple times today, but statistical significance versus clinical significance. And I liken that there's a famous breast cancer study that they looked at, and it had basically an effect of like 0.06, but it was profoundly statistically significant. And as we looked at it and we tried to incorporate all the years of data we had, you had to step back and you think, we were talking numbers needed to treat, and how is that going to impact? And while it was statistically significant, was it going to change what we did for the patients? And that really is where clinical analysis comes into play. So we've heard about different variables, and outcome variables are the dependent variables, and predictor variables are the things that we may change that will impact into the outcome. And we've heard about chance and the random error, and those are things are difficult, right? They're random, so you don't plan for them. You don't know they're coming. You try to prevent as much as you can. You try to standardize as much as you can. Bias, and Stephen talked about bias, and that's an important thing. Right? Surveillance or selection bias, how you're picking the people, how you're interviewing, all those different pieces of it. And then confounding. And confounding can really impact on your results. We're going to talk about a little study here in a second, but those are things that we have to think about, especially as we look at that lung cancer study I told you about. There was a lot of factors that they didn't take into account. They had a wonderful premise. It was a beautifully designed study, but they didn't think of all those different aspects. It's not that vegetarians could smoke more than non-vegetarians. I don't know. And that would have been related. Maybe all vegetarians work in heavy metal mines. That wasn't controlled for. So all these things can go into that. And that's where this analysis comes into play. At the end of the day, you want to say cause and effect. Now it's acute for all of us in this room because we just lived through COVID. And again, I use the banana peel thing, but we had that every day, right? The study came out, take this, and I won't go through all the ones because it makes me grayer than I am because just didn't think about some of them, but it was daily. And that's the thing, you want to look at the cause and effect and really what clinical characteristics of the association really favor the causality. So it's important that we have consistent data, and it can be consistent within the study itself, but it can be consistent over time. Now, I've been in healthcare for over 35 years, and I've been involved with cancer for the greatest part of that. But in my early days in pharma around development of estrogenic drugs, in the early days, estrogen was good. And then there was data showing it wasn't good. Then there was data showing it was good again. Then there was data showing it wasn't good. And then there was data showing it was good, but you don't take progesterones with it. And it's good, but then don't forget you can get endocrine cancers. And so that's over 30 something years. And the estrogens have been around since World War II. So it's that temporality and the consistency. And how does that data you're looking at impact and really absorb what we know from before? And that's an important thing. And I think that that's something that as you're setting up your study, it's important. That's why that first chart's so important. You want to incorporate that into what you're looking at from a study standpoint. So you're taking that into account. So you're building a more sound study as you're going forward. Different populations, that's important. I specialize in chronic lymphocytic leukemia. The median age of CLL is 72. That means half or over, half or under. Well, if you look at all of our data really in treatment for CLL, the median age, 52 to 55 in that range. So markedly younger, well, you're going to have differences. You start getting a high population of people in their 80s and 90s, that does impact on your results. So you need to understand that. You need to be able to look back. What does that mean? What about different places? Well, it's important that we have full involvement, that it represents the population as a whole. We'll talk about generalizability, but you want to make sure that that's the case. I've been part of studies that we've done where that the patient may have massive tumor lysis in the treatment, and when they opened the study up at other sites, they weren't really prepared necessarily and weren't as rapid to deal with that. And you have a death on a study, you basically have you impact on whether the drug goes forward or not. So it's those types of things that really matter, not only as you're designing your study, but as you're carrying it forward. And then the study designs, we've heard about the different studies, right? Whether it's observational, whether it's just a case report, whether it's a phase 1, phase 2 trial. What does that mean? Phase 2 trials, they have a 1A to 2, whether it's a targeted therapy versus a cytotoxic therapy. Those all matter in how you have results and how you have to look at that data. And then different times, I talked about that estrogen study, but over time, there's variations in how do we deal with those things. A lot of things go into that where smoking was decreased, where there's other aspects that may impact on results that you can't really control for. So then the strength of association, and we talked about the p-value, and that's an important thing. And though we do know that the larger the p, the larger the association. Doesn't mean it's more right, doesn't mean it's just the larger the association. And as we go forward, looking at what really matters, you're trying to take what that clinical importance is, because the counter to all this, if you think about it, and I'll look to my colleague statisticians, but for the chance that it's going to, you're rejecting the null hypothesis, and you look at that there may be somebody by just dumb luck that is going to be cured of a disease just because. Right? And one of my first patients I ever had small cell lung cancer, widely metastatic, 17 years before I saw them. And the first thing he said to me was, "You don't know that much, do you? I was supposed to die in 6 months." And 17 years later, I said, "No, we don't." Because as we say in medicine, the disease doesn't always read the books. And so it's just, how do you incorporate that? As you see those stronger associations, it does matter. as we look at that. And I would say for those thinking about the literature, you want to say it matters, and it also should matter how you interpret some of the previous data. Now, we've all probably been exposed, and certainly during COVID, you'd have people going, I just read that if I take this, that's going to help me, right? I've had it my whole career in oncology. Oh, I was listening on the news, ASCO, they said, if I get this drug, I'm going to be cured. And I said, well, it's a very small group of people. It's within the context of decades of literature. And so, that's the important part as we look at that. Associations of confounding are indirect. So they're just generally weaker than the direct ones. And that's how you want your studies to be designed. And I think that's, these are some key aspects of that. Temporality is really, it's, as it says here, the cart versus the horse. In observation, you want to see that the initial cause results in the effect, right? That that it's coming beforehand. Sometimes that's tricky because there's a lot of things that we talked about. We heard about recall bias, but if you want to find out when somebody may have started with disease, they may recall that they had it 5 years ago, but maybe it was longer, right? And there's a lot of bias built into that we need to look at. But there's tons of examples of that timing of how do you look at that, and temporality. Where are you looking at? So, for instance, if you look at maybe following up for a response and you're looking at a CT scan, you're doing it monthly. Well, you may see a small change in a month. You really need 3 months to show a statistically significant change. And if I compare point A to point B, it's just too small to really show any statistical difference, but there's a difference in 3 months. So how are you looking at that? Those are important characteristics. And it really, the key part is it has to occur, the effect has to occur after the cause if you're looking at this. So then biologic plausibility. I gave an example where they had perfectly sound reasoning biologically, but those are important, and that's really vital to do up front, right? That question comes up, and really should be thinking about all those aspects of that. And is there a mechanism that makes biological sense? And don't lose sight of that. And as you're looking at that analysis, and really the conclusion, that's going to be important. And then the gradient. Now, gradient's a little weird in that, it's not everything is dose related, right? Think about COVID. I remember in the early parts, you got to be 6 feet away, no, 3 feet, 5 feet, wear a mask, right? It was all around the premise of a dosing gradient in a sense, but that wasn't always true because everybody's different, a different host. So the gradient may be a combination of that, but those are things that you want to look at to say, okay, is this a person or a thing where just the presence can trigger the full effect? And then don't be alarmed if there's the inverse effect for certain people, depending on the exposure and what the incidence is. And secondary outcomes. And this is really an important topic in the oncology world. Back in the beginning of my career, there's a drug called gemcitabine. Gemcitabine was first used in pancreatic cancer. And they did a beautiful study on the pancreatic cancer and showed really no benefit in survival from this drug. Now, understand, in those days, if you had a 10% response rate, you probably had $1 billion drug there. But they had no improvement. But they did something really interesting. It was the first time that I recall that they did quality of life survey or quality of life secondary outcomes. And they found that people with pancreatic cancer treated with gemcitabine felt better, did better overall. Quality of life was improved. And that alone granted it its FDA approval. So the secondary outcomes, you want to align them. Now we do a lot with correlatives. So looking at biological markers for disease and for response and for other characteristics, because so many of our drugs are now targeted at specific mechanisms within various pathways. But these are important. And it's important that they align or agree with the primary outcomes. Now, the most quintessential primary outcome is death, right? Because it's a yes, no. But all these other pieces that can lead up to that are important to incorporate in how you're doing these studies, because they can be the reason that that drug has benefit for that individual person. We heard a lot about error today, and this is something that's really difficult because you step back and you think, okay, there's a lot of opportunity for error. And if you look at studies, and I saw there are a lot of coordinators here, and it's from how they're consented or not consented. Is there selection bias in who we say, well, this group is always easier to get. So all of a sudden you look and you have one population. There's not generalizability, that type of thing. So the error that you want to make sure that you're not tacitly inserting error into the study. And that's really important, not only in how it's carried out, but how it's written and how you analyze that. Because we all, and I think Dr. Ramirez said it right, that our children, the first time you go to an IRB and they say no, and they send you back, it hurts your feelings. It does. And you think like, this was the most brilliant work I've ever done. And then, same when you complete it finally, and you find out that, okay, I've got to review this properly to make sure that I am not inserting error in how I analyze this. I talked about generalizability here a couple of times, but is it broadly applicable or not? And then the usefulness or clinical importance. And that's, to me, very important as I look at this. What is the usefulness? Is it something where it's just, okay, it's clinically significant? Does it change anything? Or is it really impacted on the universe as we know it, and people across the globe will benefit from this? And it doesn't have to go that far. It could be just one piece in the journey on how we're treating that disease, but still, you want to find what that significance truly is. So I've gone through these, and we're coming up on time, but I think as you look at this, there's no one part that is more important than the rest as we look at how do we do clinical research. The more you do it, I think the better you get at it, and there's people at various levels of doing studies here. So it's important to get into that and finding your part and really trying to absorb each part of that. So as you look with the results, the statistical analysis, the clinical analysis, and then study conclusions. And I'm going to leave you with one thought, and it's something I saw on TV the other night, and I'm going to paraphrase it, but all this, all the statistics, everything else, think of it this way. It's the conclusions you make. So 44% of marriages end in divorce in this country, right? So you think a good conclusion would be, well, that means 56 and a blissful, wonderful life together, right? In marriage. That's one conclusion. The other one is that 56% of marriages end in death. So with that, I appreciate the chance for being here, and thank you for letting me have the chance, and thank you.